Hey, P-Bio, and welcome to Chapter 11, where we're talking Mendelian genetics. So it's important to note that the behavior of chromosomes during meiosis is going to allow for a lot of our understanding of basic genetics. And so we're going to keep coming back to these concepts that happened in metaphase 1 of meiosis when they lined up in double file line. If you'll remember, I really harped on the law of independent assortment. Remember, we've got like teal and dark blue chromosomes here. They could line up teal, 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 dark blue teal, dark blue, dark blue. And so when these split, these two going to this side and these two going to this side, that's going to determine the gametes. And since it'll be random which gamete fertilizes, our chromosomes can all get mixed up. And so it's this random assortment of different genes being on different chromosomes and different alleles on different homologous chromosomes when they separate that's going to lay the foundation for a lot of our understanding. And our understanding is going to go back to a dude named Gregor Mendel, who I want you to be familiar with. He was a monk in what is now known as the Czech Republic, and he grew pea plants. And he started to notice certain patterns with different traits. So things like flower color, pea shape. There were all sorts of different traits. And Mendel was doing this in the middle of the 1800s and ended up publishing his research in 1865. So what he did was he established some true breeding lines. So what that means is if you took the two plants and crossed them, the offspring always had the same traits, no matter how many generations you went forward. And then what he did was he started crossing different lines that had different traits. And in each generation, he would count how many offspring there were. So for example, one really popular one was purple flowers versus white flowers. And he would control how they were bred by cutting off the stamens and of the male plant and rubbing that on the carpel and pistil of the female plant to ensure pollination was occurring as he wanted it to. So a couple terms that I want to make sure we're aware of. So when we talk about a heritable feature, that is a character. So for example, flower color for a pea plant is a character. The different variations of that character are called a trait. So for example, one trait would be purple flowers and another trait would be white flowers of the character flower color. A gene is a unit of DNA, that little section of DNA that codes for the protein that causes that trait of the character. So Mendel in his time referred to this as a heritable factor. Remember, we wouldn't know that DNA is the source of heritability until 1944. We wouldn't even know about chromosomes until 1882. And so we wouldn't know about meiosis until 1876. So all of what Mendel is doing is before knowing about meiosis, before knowing chromosomes existed, before knowing that DNA was the heritable factors. Allele is going to be the alternate form of a gene. So this is the code, alternate forms of a gene, different variations in the letters that may cause different proteins to result in different traits. So gene is to character as allele is to trait in that finalized version. And we're always going to document our alleles as either a capital or lowercase letter based on if it's dominant or recessive. And there's two alleles to make up a gene because you have that same gene on each homologous chromosome. Sounds like a lot, but I just wanted to get that all out of the way. So some of the things that Mendel looked for. He found 7 easy to identify characteristics. So in those pea seeds, he noticed some were round and some were all wrinkled. He noticed some were yellow and some were green. On the plants, he noticed that flowers were either purple or white, that maybe they were axial on the sides or terminal at the very end. Some plants were tall, some were short. Some pods were inflated versus constricted, and some pods were green versus yellow. And after a while, he figured out that some of these were dominant and some of these were recessive. It may surprise you to see yellow being dominant considering we typically eat green peas when we go to the grocery store. So Mendel starts putting together some data, and we're going to look at two traits here: seed color, yellow and green, and seed shape, round and wrinkled. So these generations, what do they mean? P is the parental. Those were his true breeding lines. So he took one of the yellow and one of the green and crossed them. Same with one of the round and one of the wrinkled and crossed them. In the F1 generation, which stands for filial one, this is the offspring of that P generation. He found that all plants came out yellow. And in his round and wrinkled group, all plants had round P's. So then what he did was he crossed the F1 generations again. So it was just yellow crossing with yellow, just round crossing with round. And in that F2 or filial 2 generation, he found that mostly, again, they were yellow, mostly they were round, but they weren't exactly all round or all yellow. So what ratios do you start to see? Around 6,000 plants were yellow versus 2,000 for green. It's about a 2.99 to 1 ratio. Here we had 5,400 round to 1,800 wrinkled. That's about a 2.96 to 1 ratio. So these are both really, really close to a 3 to 1 ratio. For every 4 plants, 3 were the same as their parents, but 1 was the same as one of those grandparent generations out of every 4. So keep in mind that 3 to 1 ratio as we start to look at how genetics works out, and we're going to see that ratio cropping up over and over and over again. So what did Mendel conclude from all that data? Well, he said that alternative versions of genes account for variations in inherited characters. Okay, so there's different versions of genes. For each character, an organism will inherit 2 copies of a gene, or as we now call them, alleles. They'll get 1 from each parent. So we learned this in meiosis, right? With fertilization, you get 1 of your homologous chromosomes from mom and 1 from dad. If the 2 alleles at a locus or a gene differ, then one, the dominant allele, will determine the organism's appearance, and the other, the recessive allele, has no noticeable effect. So when we have purple and white flowers mixing and all the offspring are purple, we know that purple is dominant. And so these offspring are getting a purple allele and a white allele. But the white allele isn't showing up, which means purple is dominant. Then when we cross them again, we get mostly purple, but we get some white. And we'll see in the next unit kind of how that shakes out, why that is the way it is. Mendel's final conclusion was that when 2 alleles for a heritable character segregate or separate from each other during gamete formation, they end up in different gametes. So this is that law of independent assortment. Remember, Mendel didn't know about chromosomes. He didn't know about DNA. He didn't even know about meiosis and how gametes and, sperm and eggs are formed. But just looking at these P's, he was able to understand that this was going on without any knowledge of what's happening in the cell, which is really remarkable. So we can see here how this 3 to 1 ratio is forming. If you have 2 parents who each have a dominant and a recessive allele from that F1 generation, we can start to see that if mom gives a dominant allele and dad gives a dominant allele, it'll be purple. If mom gives a dominant allele and dad gives a recessive allele, it'll still be purple. If mom gives recessive but dad gives dominant, still purple. But if they both give recessive, it'll be recessive trait. So we can see how that 3 to 1 ratio starts to form. And so we can really see how meiosis is making its play here. So in meiosis, we're splitting up our chromosomes, and so we're getting 1 copy of each gene. These gametes are haploid. Fertilization is occurring, so these gametes are combining to form zygotes. And then as those zygotes grow, depending on which randomly allotted chromosomes they get, when they combine, we end up with a certain trait based on that principle of dominance. If the dominant allele is present, it's going to show up. If the recessive allele is present, it's going to show up. And so we can start to see how these Punnett squares that we may remember from genetics all stem from meiosis and that law of independent assortment, also known as the law of segregation by Mendel. Same concept. Meiosis is the key to our understanding of genetics. Sometimes we may see that there's some variations in traits. So some traits show up that are different from the traits of either parents. And anytime we see this, we're going to call that recombinant. So recombinant offspring means that we have new traits or new trait combinations that weren't visible in the parents. And that's just because of this dominant recessive going through the generations. So some terms you may not be familiar with when we're talking about how these alleles match up. Homozygous means they're going to be the same. So we have homozygous dominant or homozygous recessive. And heterozygous means they're mixed up. So if you have a dominant or recessive, that's going to be heterozygous. And that comes from the Greek homos and heteros. So two other words I want you to be familiar with. Phenotype is going to be what we see. So like round phenotype versus wrinkled phenotype. So I remember this as phenotype describes physical trait. Physical trait starts with pH. Phenotype starts with physical or pH. And then the genotype is what are the alleles you have. So if you have dominant, you would have a homozygous dominant genotype, heterozygous genotype, homozygous recessive genotype. I remember that genotype is what's coded for in the genes. Sometimes we don't know with the dominant trait if it's homozygous dominant or heterozygous. We just don't know. So one thing we can do if we know the phenotype, but we're not sure on the genotype, is a test cross. So what you do is if you have an unknown dominant, cross it with a homozygous recessive. If all the children are dominant, we know that it was homozygous dominant. If half the children are dominant, half are recessive, we know that it was heterozygous. Now, this works great with plants or insects or bacteria, not so great with humans. Do not try a test cross at home. All right, we'll check in next time for 11.2.